I always wait for Janet to start recording. Oh, there we go. Okay, so hi everybody. I'm Kevin Sykowski from the University of Toledo, and this is our 9th webinar. I think it's our last webinar for this academic school year. I always share my screen and just show you some stuff here. So here's our title for today. Yell if it doesn't show up, right? So GLOBE Greenup is going to be presented by Boston University, Kathleen, Don, and Peter. And just a few things about GLOBE Mission Earth. So this is the group, we're the group putting on these webinars for GLOBE and NASA. We're a partnership between the University of Toledo, Boston University, Berkeley, UC Berkeley and Wested, Tennessee State, NASA Langley. We also have the state of New Mexico and the Palmyra Cove Discovery Center are also now partners. And basically we're trying to reach K-12 teachers and students, but also citizen scientists and college students to engage them in GLOBE and through NASA resources to do projects. And I don't know, this was a question I had. Are we going to try to do a student follow-up on May 18th? I see Kathleen shaking her head, maybe yes, tentatively. And Janet, I don't know if this is the link for that.
It should be.
Okay. All right. Oh, great. So we'll have a student follow-up on Friday at 1 p.m. Eastern Time. All right. So I'll turn it over to the Boston University group. Let me stop sharing. Hey, it worked. It doesn't always work, so.
You're not supposed to sound surprised, Kevin. You're supposed to, you know, that always happens.
No, I would say we're 50-50. My computer is getting old. So Kathleen, take it away.
All right. Just making sure this is sharing right.
Oh, it looks like it's sharing, so that's good.
There we go. All right. Well, welcome everybody. This evening we're going to be talking about Spring into GLOBE. So talking about GLOBE Greenup protocols. We have a few that we'll go over, and really what we're looking at are the plant observation protocols and investigations and some of the lessons we've learned from the grades 2 through 6 classrooms that we've been working with in Providence, Rhode Island. My name's Kathleen Johnson, and I'm currently a doctoral student here at BU and part of the BU Mission Earth GLOBE team.
Don DeRosa, I'm a faculty member here in Science Education and part of the GLOBE Mission Earth team.
And I'm Peter Garik, and I'm also part of the GLOBE Mission Earth team.
All right. So our agenda for this evening, we want to talk about GLOBE in and out of the classroom, and what we mean by that is looking at the protocols that we can use inside of the classroom, so sourcing GLOBE protocols that can be done within the classroom, and then looking at those GLOBE protocols that take students outside to explore nature and outside.
Sure. Yeah, and these protocols are protocols that we've used working with the students in Providence, Rhode Island, and they're meant to be, at least the way we've used them, is in a progression, and that progression is shown in those bullets, and we'll present it in that fashion.
Go ahead.
Okay. So we do have time for questions and discussions afterward, but to really make this a very productive webinar, we welcome your questions anytime during the webinar, and we'd love for you to share your experiences, your thoughts, any of that. So we are hoping you will spark the webinar fuse.
And avoid the webinar snooze. We thought that was clever. I don't hear a lot of laughter.
You don't have to be on mute right now.
All right, that's funny. I like it.
Thank you.
Thanks, Kevin. No, but the point here, though, is we want people to feel free to participate, to ask questions. And anytime we do any type of a workshop or seminar, if you will, or webinar, it's real important that the richness of any of these experiences is not necessarily sitting among these three people, but it's in the people that are participating. We know there's a lot of experience, a lot of insights, a lot of knowledge. And so we welcome people to share that. That's really what makes these types of, I think, experiences rich.
So can I take you on that very kind offer?
Sure, please do.
I believe, Kathleen, you are the PhD student in VU?
Yes.
So in the regards to this, what was this topic of your research? What was the actual objective? And
so the topic of my research is largely, that's a good question. I am looking at science education focused largely on deaf learners, but really my focus area is curriculum and teaching. So that is where my interest is with GLOBE, and this project and what I've been doing on the project really supports my research in helping me hone my skills in curriculum development and in teacher support.
Kathleen has become proficient in her years here at ASL. I'm really impressed. And she's working very hard to incorporate and integrate STEM and those investigations associated with STEM into the ASL community. So it's not too many people working on that, and it's we could get into the background on it, but it's something that's sorely needed, I think, and it will certainly address a large gap in this.
absolutely. Thank you for sharing, and please go ahead. I apologize for interruption, but you invited, and we are not snoozing here. We are
Good, yeah, good, no snoozing.
No need to apologize whatsoever. Thank you for the question.
absolutely. Thank you.
Okay, so just kind of giving an overview and purpose of what we do.
So, yes. So, as part of our NASA CAN, what we are trying to do is develop ways to integrate GLOBE and NASA assets into the K through 12 curriculum. Much of what you're going to hear about today is focused on what we've been doing at the elementary level. We're working in Providence, in urban elementary schools, with a diverse, underrepresented minority population, and also with limited access to the outdoors. In fact, you can see here in these pictures the green area that these schools have available to them. So the question is, how do we introduce to these students GLOBE protocols that are based upon how growing things grow, how living things grow. And as it turns out, these are of particular interest to the teachers, and we have found quite a bit of interest in following through with these.
Okay, so so if we're looking at the demographics of the schools that we're looking at in Providence, this kind of gives us the snapshot.
Yeah, this is representative of the district and of the students we're working with. So this gives you some idea of the diversity in the schools, which is a rich diversity. We see that as certainly a positive for us.
And furthermore, this of course does represent a good portion of urban America and the nation that we live in, and therefore working with this a student group and learning what we can do is of great value.
So I have a question for all of you. How many of you have heard of or are familiar with bottle biology?
I think I am.
Yeah. I think we have to go with yays or nays because we can't see you.
See anybody. I don't know if I shared my screen wrong or...
People are putting their comments in the, comments. So yeah, Marcy said yes, Tracy said yes. Jim said no, Joe yes, Jeff something similar, Steve yes, Larissa yes.
Okay, so there is some prior knowledge here. Great.
Wonderful.
Yes, Susan yes.
Okay, so yay, that's wonderful that there's prior knowledge and some familiarity with bottle biology. What we were looking at with the bottle biology program, we made some adjustments to it, but still following the overall purposes and what students are doing using this GLOBE bottle biology. So the protocol is providing students an opportunity to study plant life cycles any time of year. And really, with the way we have our protocols and the protocols we're going to talk about, this is a really good kind of get your feet wet experiment for the students, to where they can become familiar with plant life cycles. They can become familiar with what plants need to grow and with what plants need to reproduce in this closed setting of the classroom over a fairly short period of time. So they're not having to wait for the full spring to fall cycle to observe this, but they can observe it in a lesson in the classroom. And then also they get to link what they observe in this controlled setting with some of the other protocols, the phenological protocols or phenology protocols that GLOBE provides, and that we'll also cover in this webinar.
So in this picture you can see the flowering of fast plants in the window of a third grade classroom in at the Webster Avenue Elementary School in Providence.
And I will say the students are just having a great time with this. They're really excited to share what they're seeing. So this is another third grade classroom at Fogarty Elementary School in Providence, Rhode Island, and showing what this teacher, Ms. Pimentel, did to set this up. So we do have a video walkthrough on how to set up the systems for bottle biology, and Ms. Pimentel took that video and created a checklist with it for her students to follow to make sure that they were executing every step. She also had her students working in pairs, so you can see a young man in a white shirt and a young girl in her little blue shirt. The girl is holding the top of a bottle, and the young man's getting ready to put some soil in there. And then she had her students set their bottles in really nice, secure containers to put in the window so they wouldn't get disturbed while they're letting them grow. All right. So some of the questions that we can explore with this would be, how do plants grow? So looking at life cycles, what do plants need to grow and survive, and how do plants reproduce? Looking at their growth and reproduction.
Yeah, and we do feel it's important, and we're going to leave that a little bit too, but whatever the connection is, the hook is, that it invests the students in this, so that gives the students purpose and meaning. And some of those questions are just questions, examples that one might use to hook the students, to make this a meaningful exercise, so they're figuring something out, if you will.
All right, so the way we adapted the GLOBE protocol, we actually chose to focus on Wisconsin fast plants, a type of mustard, instead of focusing on the radish plants.
And that's because when we tried the radishes, while they grew very spindly and they would not flower. Getting radishes to flower is difficult, and therefore showing the entire life cycle is difficult.
Now, is radish a 2-year plant? Or does it, oh no, it flowers in 1 year, doesn't it?
In principle, it does. The flowers are very small, and getting the flowers to, in fact, bloom is not easy. And having the flowers is a draw when you take a look at the second grade standards, because the second graders study pollination and pollinators. And so being able to have flowers is of value to them.
Okay.
And really quick, I'm going to stop screen sharing because I want to show you some of these plants. They aren't in a bottle biology system, but they are the Wisconsin fast plants, and you can see those bright yellow flowers. To see if you're wanting to explore reproduction in the classroom, all students need is a magnifying glass to see the stamens and styles in these. And then they produce fairly long and really obvious fruits. I don't know if you can see those on there, but it's basically like a miniature little pea pod, that little spike sticking up there. And when those are mature, you can see the little bumps in there that allows students an opportunity to interpret, what do you think is inside of that? What do you think is going on in there? So with this plant, there are a lot of opportunities for students to make predictions and make observations in addition to following the plant life cycle.
But we also consider this the first encounter in our progression.
Mm-hmm.
So we're not going to get too complicated. This is the first opportunity for students to kind of get familiar with what somewhat rapidly growing plant that they can see over a reasonable period of time. But we're not going to get too deep. They're going to get familiar with some of the things that happen to a plant. We're not going to get into a lot of terminology about blooming, about leafing, and so forth right away.
Okay. Did it work? Is it sharing?
Are we back on screen sharing?
Are we back on screen share? Can you all see our PowerPoint? Oh, how long from seed to fruit? It's 35 days, so it's fairly short. And that is actually from seed to a fairly ripe fruit. If you go 40 days, then those seed pods are definitely completely nicely dried out, and the seeds can be harvested.
And yes, we see your screen.
Okay, thank you.
Thanks.
So the pictures on this slide are some more examples from classrooms and what they've been doing, some of the additional notebooks and activities that they've used. But with the adapted protocols, some things that we haven't tested yet, but that can be extensions and experiments on this, besides just planting the basic setup, would be to plant an additional setup with limited resources. So no water, no nutrients, no light, no soil, and having students make predictions on what they think might happen with their plants in these altered conditions. The other benefit to using Wisconsin Fast Plants is that there are multiple varieties, so students can study variation within the fast plants by planting different types of seeds, and again, making predictions and making observations, comparing how these different varieties grow. Other suggestions to extend, students, we do provide, it's really funny what we wound up doing. So our bottles, we're using the half-liter water bottles, so it's a fairly small space. So we used some of the little the wooden coffee stir sticks, and marked those off at centimeter increments for students to be able to use as rulers and measure the growth of their plants on a regular basis so that they can potentially graph growth rate. They can also record how long it takes their plants to flower, how long it takes their plants to produce those seed pods. With the data recording, they can compare their data with other classes. They can compare their data within their classroom. Because these have such a short cycle, they can collect the seeds and actually replant them and compare that next generation with the parent generation. And then they can also look at pollination. They can use paintbrushes or bee thoraces to pollinate the flowers and see if maybe there's a difference in seed production. Are there any questions? The Wisconsin Fast Plants, so the question is, do we purchase the Wisconsin Fast Plants for the teachers or use the seeds from previous years? We have purchased them for the teachers and supplied 4 seeds per student.
They're very small,
yes.
for little second graders' hands, so it's good to keep them you know, on a nice white piece of paper and to keep them organized. It's not always or even tape is helpful.
Yeah, so on this slide, there's actually the little white card there with the little black spots. Those are the seeds. But looking at some of the lessons that we've learned, we did make a video, and I'm going to show you like 2 seconds of the video really quickly.
So this is available on YouTube. the teachers have been showing this video to their students prior to their own planting.
And is it playing? Can you guys see this or no?
Yes, we can.
Yeah, because I can see it on this one.
Yeah, thank you. We can see it.
Okay, so I'm not going to play the whole thing because it is 10 minutes long, but as Peter said, the teachers can use that prior to setting up. What we did learn, though, is that it is really important to have a checklist. So we do have an image-guided checklist. This is our third iteration.
Yeah, we've learned, we started off with a bit of a complicated checklist that was all text. Of course, that doesn't work. And then we simplified it, and we found out that what's really important, we should have known this, right, is to have a little bit of text and some pictures.
Yeah.
And it's certainly good for students with, our ELL students and all students. It's universal design, but we found it's very helpful to have both of these. And the students can follow that as they go along. And even with that, of course, they'll need some direction.
Yeah.
But at least they have some guidance.
So these are available. They're available, obviously, on the hot links on this presentation, but they are available to all the teachers in both a PDF format and a Word doc format, just in case teachers want to download the Word doc and make any changes that are applicable to their own classrooms. It's really important to keep the reservoirs of the bottles filled with water. If they dry out, then the plants dry out, and it's pretty much impossible to get them back. And then tips on seed collection. The seeds can spray everywhere if you just pop the pods open. So putting them in one of these little plastic bags and sealing that and then just kind of rolling them around actually does a good job of releasing.
If you haven't seen the pods, they remind me of little string beans.
Yeah.
Little string beans and inside of the seeds, but they're just very small.
All right. So this is largely an FYI slide to let you know what is addressed or how to approach three-dimensional teaching using the GLOBE bottle biology protocols and what cross-cutting concepts, science and engineering practices, and disciplinary core ideas are addressed. So I'm going to give you just a minute to look through those and let us know if you have questions or comments.
There's always a bit of a caveat when we talk about NGSS and any standards, because no one wants to claim that any one experience or lesson are going to hit all of them. But there's certainly opportunities for these to be addressed in this progression that we're talking about.
Right. And so the next slide, a question that comes up with most teachers, as it should, would be the science standards that are met. And so this is a list of the science standards that we've identified that bottle biology meets. And a really good point that Peter made with this is looking at it, it does address,
question here is how is LS3, A, and B addressed? Or how are they addressed?
Inheritance and variation of traits. So this would be if you do choose to harvest those seeds and plant that next generation of seeds, students can compare their data from the first generation to the second generation. In the world of technology, having students in some classrooms take pictures, cell phone pictures, or any other resource pictures is also an option, and they can compare images, but making sure that they're keeping really good notebooks, so not not completely overwriting a notebook with pictures, but being able to compare the traits they observe in that next generation.
So in particular, with the fast plants, it's possible to order the seeds with different traits. So this is an opportunity to do a genetics experiment to see what the inheritance is of the different traits.
Mm-hmm. And I think, too, it's important to note that that sounds like quite an advanced investigation, especially for elementary school students. But we have to, this is a progression, it builds. So even if they were to take the first time or the second time they grew these plants, if they were to take and plant the seeds they harvested, then had a chance simply to compare them to the seeds, the parent plants. We don't have to necessarily go through Punnett squares and inheritance patterns, but just for them to realize that there is an inheritance going on, that they do look like the offspring do look like the adults and sometimes don't look like the adults. That's all groundwork for more, for a deeper understanding of genetics. And that's part of what we think we're doing in overall Peace and Mission Earth, is that these are progressive. What students do each grade, they build upon each other. And so we're just spokes in the wheel, if you will, but they're interrelated. We can work together to build those foundations.
And another key point with this is that it is an indoor investigation, so it's something that can be started at any time of the school year, depending on how many extensions you may want to do with this, that it can be started early and continued through, just depending on what the goal is.
Yeah, Jeff, you can work on fast cilantro if you want. That would be a great discovery. I have to explain to Peter the question. He doesn't see me.
Oh. So with the actual standards, a really awesome thing about bottle biology is that it does address standards for levels from kindergarten all the way through middle school, that it can be used in all of these grade levels. And we were discussing before this webinar about how students can do an experiment in 3rd grade and then be doing another bottle biology experiment in 4th grade and be, looking at that data from 3rd grade and comparing to previous years. It doesn't just need to be within the same year.
Yeah, that school kind of builds up a legacy of their own data for that site, which is something, of course, GLOBE has been promoting for years, right?
Okay. So I fixed that. The next protocol that we're looking at is another indoor protocol, and it's one of the GLOBE Greenup protocols, but really kind of a zoomed in version, and asking a question about what happens to trees in the winter, what happens to these leafy trees that lose all their leaves in the winter. So we've called this Alive or Dead, the Mystery of the Winter Twig, and it's really taking that GLOBE Greenup protocol and honing in on these twigs and having students make observations and ask questions about the twig to try and decide and make an informed decision through evidence. Is this twig dead or is it actually alive?
So the idea here is that you would really like to have the students have a context for what they will be doing when they go out to a phenological garden and take a look at the lilacs and the viburnum. Just going out and looking at the buds on the lilacs is not enough of a context for understanding what's going on. Mm-hmm. So by having the prior activity within the classroom that we're now going to describe, we've set the context and engaged the students in taking a look at what happens outside.
So the learning outcomes for this are very, very sort of straightforward. We want students to think about and try to, the phenomenon, if you will, is these twigs, are they alive or not? And then the students need to explore that a little bit and find clues, any evidence they can for those twigs being alive. So it sort of sets the stage. They will find out, of course, that they're alive. I don't mean to spoil. Is that a spoiler? Right. I'm sorry. Yes, it's a spoiler. No. But they need to.
You're not supposed to give the answers.
This is something that I find interesting. We have a butterfly bush at my house. Uh-huh. And is it alive or dead? And it's hard to tell, but there's no buds on it now, so I'm thinking it's dead. Or the main shoot dies and then it starts growing from the ground again.
Well, you can conduct this little investigation, Kevin, when you go back. Just hang tight and see what's going to happen.
Yeah, but anyway, this is an interesting question for students, right?
Yeah. This didn't change.
Yeah.
Yeah, it was. Sorry, technical difficulties for just one moment.
But Kevin, you bring up a really good point. I think that's what's engaging, and we feel that, especially we learned that for these young students, that if the question is straightforward enough, they can spend their time really trying to figure something out without having to try to figure out the question. Rather, they can start to work on the content and the topic. Okay, go ahead.
So with the Alive or Dead Mystery of the Winter Twig, one of the critical things to bringing this into the classroom, and Dawn has pointed out many times that it will go outside of the classroom as well. But first step, bringing it into the classroom, students need to be looking at these dormant twigs. So the image that I have on this slide is showing those really dark, well, those solid brown scales. I shouldn't say dark, not all of them have dark scales on the buds, but if you look at those things that look like little thorns on the tips, those are the dormant winter buds. And I know for me as a kid, I had no clue in the winter that there were actually buds on a tree. I just noticed that it died in the winter and came back to life in the summer. So again, with the green up investigations straight out in the garden, if we're telling students to go look at the buds on a lilac bush, they may not really be solid in what they're looking for. And this gives them that opportunity to look at this up close and not just look at it, but to pull it apart. So the students are going to be exploring and making predictions about the twigs before they even dive into them and start tearing them apart. And then they're going to be able to link what they're observing in this project to what they see in the phenological gardens in the next protocols. Yes. Okay. This is actually a picture, oh, and I didn't label it. It's from Mr. D's classroom. What grade? Mr. D at Carnival.
No, Carnival. So this is 5th grade.
Yeah, so this is a 5th grade classroom, and the twig you see in the background is one that has greened up. But what we're doing is we're bringing these clipped twigs into the classroom and starting with a hook. So we had planted these phenological gardens in Providence, Rhode Island, and when I did this in a 3rd grade classroom, the hook that I used was actually the phenological gardens because the students had helped plant the gardens. So I came into the classroom and I told them I was really excited about your gardens, and I went out there and now I'm really worried because it looks like all of your shrubs are dead, and showed them a twig. And I'm like, wait a minute, but you guys can't see this twig, can you? And they are all shaking their heads. And I told them, that's okay, I brought enough for everybody. Pass them out and have them draw the twigs and investigate the twigs and collect evidence on whether this twig was alive or dead. The materials are the twigs, but some extra twigs to be able to put in water after the students have dissected theirs, so students can actually see these twigs green up in their classroom and record the data.
But prior to that, don't you have the other slide with, do we show that?
That one?
No, they split it open. Yeah.
That's next. That's close.
Yeah,
okay.
Yeah.
The other thing that we've done with this is we used a really simple data sheet. And honestly, the data sheet that I made was not a simple data sheet. The data sheet that I made was asking these same questions in a very long form. So I was asking students, what evidence can you find in your twig to suggest the twig is dead? What evidence do you find in your twig to suggest the twig is alive? Based on your evidence, do you think your twig is alive or dead? And then Dawn came in.
Yeah, and my feeling is probably because I have a very simple approach, because I'm simple in many ways when I try to look at things. But said, we're telling the students way too much or giving them too many hints. Give them the opportunity, the freedom, the license, if you will. And for those of you that teach elementary, I know there's guidance that's needed sometimes, but in this case, we simply ask them, do you think it's alive or dead? What are your clues? You figure it out. And that's part of what we do with phenomenological teaching. Ask the students to figure it out. If they need guidance, we can give them some hints, but at first, give them a license to think a little bit openly and freely. And if you're familiar with Paige Keeley et al., who writes a lot of these probes and assessments, this is sort of like that. They put down their evidence of it being alive or dead. Then we ask them simply to make a choice or make a claim. Is it alive or dead? They circle one or the other. And then they can use the clues, which are right above, to explain their choice, or to justify their choice.
And doing this in the classroom, it was so much fun the moment students asked. So they actually didn't start dissecting their twigs on their own. They raised their hands and they're like, can we break these? I'm like, break them, bend them, pull things off of them and see what you find. And that was one of the richest moments in the classroom where all of these children are going, whoa, whoa, there's green. Oh, I see green. Look, this is green and this is green. And so they found this on their own, and they made these discoveries on their own, and they were so excited to share this. Look, if I dig under the bark, it's green, and look, if I break open this bud on the end, it's green. All right. So the next phase of this, we're not quite done with the mystery of the winter twig. We kept those, we wanted to keep those extra twigs to put in water, so that students can actually watch this process, but also so that they can get used to using the data sheets and filling in the data sheets. And so this slide shows the Budburst protocol data sheet from GLOBE and the questions that students would be answering. And they get to start and they get to practice right in the classroom where they have teacher support and where they can be watching this on a daily basis and actually really see those changes firsthand.
So we can consider this essentially readiness training for doing the Budburst protocol outdoors, and being able to catch the right time for when either the lilacs or the viburnum, in fact, do show budburst.
And the big thing, I think, one of the things here that I think is notable is that we're asking students to predict, to make predictions, now that they've hopefully discovered that there's evidence that these are alive. And there's maybe, if you will, miniature or baby leaves tucked away inside these buds. Predict what will happen when we put them in water in a window. So they're making predictions based on their evidence, and this is an important component of the practices, the science practices. And we want students, even at the elementary level, to be doing that with something that they're invested in and they have some ownership. And it's easy enough for them to make a prediction, but it's still a very rigorous and robust prediction. So we're finding that with a lot of the GLOBE protocols that this ramping up, if you will, for elementary school students is helpful.
Mm-hmm. All right, so now we want to test your Budburst brain, matching an image to its description. And so I'm just, let's see, I can see a few people, but I'm just going to ask you all what you think. How many people think the image A is a dormant bud? How many people think image A would be a Budburst bud? Full leaves?
Budburst.
Which one do you think is Budburst?
I thought you were saying A.
Yeah. Do you think it's Budburst?
I was.
Okay.
Kevin.
I see thumbs up.
Okay, thumbs up. Awesome. And then C, what do you, do you think C is dormant, full, or first leaves? We'll just go with these two. So is C dormant?
Jeff is asleep, so it must be
okay.
dormant.
Oh, no.
Well, we'll jump to the answers. So
Webinar snooze.
right?
No, that's the point. Is dormant.
Okay. A is
sleep.
Budburst, B is full leaf, C is dormant. Awesome. Yes.
There are your answers. I'm sure you got them all right.
Dormant,
Everybody did.
your Budburst, your first leaves, and your full leaves. I will say that not all shrubs have a really obvious budburst. So this is a picture of a lilac budburst, and it's beautiful and vibrant and bright green. Oak trees have this minimal little bit of swelling and then a little bit of green, and then all of a sudden it's just pow open. So again, making these observations in the classroom allows students to notice that little bit of green, that there's a change that happened, and instead of possibly missing that outside. So the next protocol that this links to is really taking it outside. So we've done this in the classroom. Now we're going to take it outside and observe our plants in the garden. For the gardens that we have established in Providence, the shrubs we've put in there are Lilacs and Viburnum. The common name of the Viburnum we used is Southern Arrowwood. And students get to use the same data sheets that we've looked at, taking those outside and recording these naturally growing plants outside and seeing naturally when these plants are actually greening up, when they're going through budburst. GLOBE is using some of this information to look at some effects of climate change. GLOBE is also closely associated with a group called the USA Phenology Network or Phenological Network, and they are using the same type of data to see if global climate change is affecting green up times and green down times in plants. So when students upload their data, they're actually putting it into this larger database, sharing information that's being used for a larger purpose.
And again, I'd like to emphasize sort of the progression of conceptual development here. They've had a chance to encounter already and think about leafing out and bud bursts and so forth. They've seen it before. They might not own it completely yet, but they're familiar with it. So when they get out into the garden, they can focus a lot more on what's happening there than learning these new terms, if you will, or learning getting brand new experiences. They have a good idea of what they're looking for. Now they can apply it. So they can start to think more about the content a little bit more in depth rather than having this be new to them all at once and so much.
And they have their firsthand observations as well to ask themselves questions when they're looking at something and maybe they're not quite sure, okay, is this a first leaf or is this bud burst? They have those firsthand observations to refer back to on what they know. Okay, well, do I see an actual leaf or is all I see green? So they have that experiential information, which that experience is really important.
And to self-correct as well, because we have to make mistakes, and students can make mistakes along the way, but the third or fourth time, you start to get it. That's the way we typically learn.
Just a quick question, if I may. Do you have any examples of data visualization just to illustrate the whole globe picture and how it really reflects on just seeing it in general? Do you have visualization?
Data
visualization.
Not currently, but after the webinar, I could probably find.
That would be lovely. Thank you. Yeah.
Are you thinking of graphs or just?
Whatever form of visualization you might want to refer to, that would be really great. Thank you.
Okay.
Sure. Yeah. No, that's a good question, and I think that would be very helpful.
I can absolutely find that and share that.
Thank you very much.
You're welcome. Okay, again, they're going to be using the same data sheets, and this is an image that, again, I didn't label. But this is students at Webster Elementary School exploring their garden and collecting data from their garden, collecting green up data from their garden. And then looking at the lessons that we've learned, this lesson, the Alive or Dead lesson, is a great 5Es learning lesson. It's a great phenomenological lesson where students encounter a phenomenon and explore that and develop their questions and make their predictions before we're really heavy on looking at technical terms, if that makes sense.
Yeah, we've learned, I think, both that with the first time the teachers are experiencing it, and it's also the first time the students, and the first time we are too, in some respects, is to keep it simple and let it ramp up, and be patient and let students build that up. So for instance, when we go out into the field or into the gardens, rather than go through a complex explanation of what students should be seeing, we call that shooting on them, or they're shooting on themselves. What should we see? We want to keep it very simple. They've seen now bud burst at least in one form. So go out now in the garden and find evidence, if you can, of bud burst, of first leafing out, perhaps. Any flowers as well. Do you see any blooms? And it's amazing because the students go around. They see things that I would never have noticed. They saw the magnolia tree. Yeah, I didn't even notice this. They had flowered already, and a lot of the petals were falling off. They saw little shoots coming out of that tree, right out of the trunk. They asked, is this a leaf? And sometimes basic questions, they're asking, how do I know if it's a leaf or a flower? It happened to be tulips. That was a great opportunity. Who knew? We assumed that they would be able to tell the difference between a leaf and a flower, but indeed they didn't. So they raised a lot of questions, a lot of insights, and I was pleasantly surprised to see how invested they were in asking questions and also making discoveries. One young person said that, this is like us. It's like when you're born, it's when there's the bud burst. When you're growing up and a teenager, it's like when they're at the first leafing out. And then when they're all leafed out, then it's like we're grown up.
From my perspective, among the lessons learned is how useful these kinds of projects can be in multiple grades at the elementary level. They connect nicely to the NGSS, and furthermore, they can embed GLOBE within other activities that teachers need to engage in order to cover the standards. So that way, GLOBE now can become part of a regular set of lessons that covers more than one topic. The students just are immediately interested in anything that's growing and in plants. So we have both teacher and student response to these activities.
The students are so excited to share what they
notice. And when they go outside, the students don't just look at the phenological garden that we've planted, they start looking at the other plants that are around them. And questions then arise about habitats for insects. Again, a topic that is not exactly a GLOBE topic, but is one that fits within the NGSS. And therefore, the entire connection begins to emerge where GLOBE-oriented protocols and investigations now become connected within the curriculum for K through, at least what we've seen, K through 6 instruction.
So with both of these, again, looking at the standards, we're looking at them covering quite a few disciplinary core ideas and science and engineering practices, as well as touching on some of the cross-cutting concepts. And then these are the standards that we were able to identify that these align with. So briefly, the last protocol is Phenology, and really it is very similar to the Greenup protocol, but instead of just looking at shrubs, at trees and shrubs, students are now looking at wildflowers as well, and they are using the longitudinal observation of data to look at when does a flower show its first leaves, or when does a flowering plant show its first leaves, when is the first flower produced, when is the first fruit produced, when do the flowers start to drop, and when do the leaves start to drop. And so really observing and collecting data on these cycles through the season, but not just on one plant. They're looking at multiple plants, so they're able to compare data between different plants. And using the GLOBE website or using one of these other two websites, one of them is Budburst and the other is Nature's Notebook, students can compare their data between classrooms. They can actually even compare it globally. So those two websites that I just named were developed by the USA Phenological Network, who GLOBE has developed their protocols with as well. When we're looking at GLOBE protocols, and then looking at the protocols between these two websites, they're extremely strongly aligned. And GLOBE even talks about the USA Phenological Network in their Lilac protocols, and refers you to go to the USA Phenological Network site, which is actually how I found these.
Good. Yeah.
Good?
No,
okay.
Nice.
I won't spend too long on these because I know we're running out of time here.
Kevin, how close are we to cutoff?
Well,
Time.
usually try to end by 9 o'clock, and
okay.
sometimes we'll have some questions and answers, but anyway.
So with these, they really do, with the Phenology protocol, it really does follow the same pattern as we saw with the Greenup protocol, but again, focused on wildflowers and herbaceous plants. What we learned with this, again, is similar, that the students need to become familiarized with the data sheets. So referring back to both bottle biology and Alive or Dead, these have given students the opportunity in class to see these phases that they're looking for, to see these structures that they're looking for in the classroom. Now they can take that outside and look for these things in their gardens, see how a flower is similar to what they saw in their bottle biology and how maybe it's different from what they saw, or see how fruits may be different between the various plants. And again, the Phenology meets a lot of the science standards and covers from kindergarten through middle school standards, or addresses, I shouldn't say covers, I should say aligns with these standards. We wanted to end with a summary of the lessons that we learned from using this progression or looking at this progression. And something that Dawn mentioned earlier is starting simple and building from there has been really productive.
Yeah, we found that these protocols really start to take traction when we give students and teachers and ourselves, of course, working together, the time to, and I've used this term before, to begin to ramp it up a little bit, to take our time, to give students opportunities to become familiar with whatever, in this case, with phenology, but with the components of phenology, if you will, with blooms and leafing out and budburst and so forth, and growth in plants, to give them that opportunity. And then when it comes time to really delve into some of these protocols in depth, the students are more ready to do that. So we found that is a take home for us in any case. It might be self-evident to all of you, but it's something that we found is really, really helpful, especially when we put in practice.
So why does it say student-centered what?
Oh yeah, that was me, sorry.
Well, we were kind of wanting to get y'all's take on what student-centered means.
So we're thinking about, student-centered, that term's thrown around so much. And again, I'm sure you all have very good insights about that. We're beginning to we're realizing, although we use the term a lot, we think we know what it means. But again, you begin to learn different things. Our take home from that is that student-centered certainly isn't something where the students drive the curriculum on their own. It's very much planned spontaneity. We create an experience, a context, and the teacher, I think, has to be very aware of where we want to go, what our learning outcomes will be, and set up an experience so the students start to raise those questions, and we're pretty cognizant of the questions they're going to raise. Mm-hmm. But the student should walk away saying, hey, that was my idea. I thought of that. Even though we anticipated and we sort of set them up for that in a good way. So student-centered, I think, in many ways gives us the opportunity to let the students drive it with training wheels, so that we're able to recognize, hey, you know what? A student just brought up something about differentiating between flowers and leaves, and we look at the tulips, and they're asking now about what these parts are, the stamen and pistil, though they don't know what they are, what they're for. This is an opportunity. I know we're going to address that. Here's an opportunity now to strike while the iron is hot, and I've prepared that, but now I can launch into that right now. I don't know if that makes sense, but that's what we're learning in terms of what student-centered really is. And if any of you have any ideas about that, please feel free to share.
Yeah, excellent. Just a quick question. This is Larissa. Have you brought up the aspect of climate change in this project, or you feel is just a little bit beyond the frame?
Well, climate change, of course, is the motivation for the phenology study. And we didn't mention it, but that would be a motivation that we would suggest to the teachers, that after they've done their budburst activity indoors and they're going outside to make measurements and observations of the lilacs and the viburnum and reporting that data, that the rationale for reporting it is exactly this issue of climate change and how things are becoming warmer earlier in the year.
And then with this legacy of data, that also links to that, where students can be looking at their past data. They can be looking at past data from years back and comparing their data on when in the season green up started compared to the year 2000. When did green up start? And see if they're noticing trends of change, and then looking sequentially through the years and seeing if they can track that change.
Yeah, but I think that's the big question, isn't it? I mean, we're really preparing the students to be able to understand and appreciate in an evidence-based way what climate change is. That's sort of the end game in many ways, if you will, and then to understand a lot of the principles and components behind that. So we haven't gotten there yet, and that's a really good question. But I think that's really you bring up a good point. That's the end game. We want students to be able to, when they do start to think about that, think of it, because now part of their being, in a sense, is looking at evidence and seeing how the evidence informs their decisions, rather than a faith-based, hey, climate's changing because we told you it is, and we're your teachers and we're the experts or whatever. You know, we eventually want to ramp up to that, I think.
Sure, great. And just a quick follow-up. Was it difficult for the students or teachers to upload the data to the GLOBE?
That's a good question.
It is, quite frankly, right? We're finding that's a challenge.
We're still working on that.
Yeah.
Is there anything that GLOBE team can do to assist you and similar groups in learning how to submit the data?
I think I need to do some more self-familiarizing with it, and then develop some questions around that. So I want to explore it further and take the notes that I take on areas that were easy and areas I was struggling with.
This year was a difficult year, as you probably know, here in New England, because of the changes in weather. And as a result, the teachers were not able to get out with their classrooms to clean up the gardens at an early time. The outcome of that was we didn't have I would say we weren't coordinated enough to ask the classes to go out and do their observations. We actually got to the Phenological Gardens before the teachers were able to, for the lilacs, to observe the Budburst. I don't know, we have one Providence teacher on. Sue, are you listening?
Eileen.
Hi, would you like to tell us something about the use of the Phenological Gardens? Oh, and Eileen, you're on also. Sue, anything you would like to say?
Experiencing the gardens with the children is great, ongoing, really good. They're enjoying it. We're seeing the growth. The weather did hinder us from getting out there, but we're going out there now, and they're really enjoying it.
Okay, and Eileen, I see you're on as well. Would you like to share anything? I don't know if you can hear me. Oh, well. Okay.
Let me say that one thing I really appreciate, is the teachers we've been working with. It's been fantastic. We go out there in the garden. I feel like, pardon the pun, but all I have to do is like plant a little seed, and they're so good. They're the educators. They're the experts, and they run with it, and they do such a good job of inspiring their students and asking the questions and following up and doing the science circles. I really enjoy working with them. I think it's because of them, a nice partnership that's evolved. They're so good being able to take the lead. We just have to put in a little spark, and they ignite it. It's really good.
And in the classroom with the bottle biology projects as well, I would say the same thing.
But Larissa, I think, if I may, I think that you bring up a really good point about the data entry. That's been I don't know if other people have had that same experience. I don't know what the answer is, but I think sometimes if there's a quick start guide, a way to, when things are easily accessible, especially at the beginning, maybe if there's a minimum amount of data to begin, to open the door, the window, if you will, and then people can become more familiar with it, and then we're more apt to put up more in-depth data. And I think GLOBE does a very good job. I think it is, quite in many ways accessible, but in many ways it's not happening with us in any case. So I don't know what you raise a good point.
Thank you. I just would like to congratulate your whole group with an outstanding job, and teachers who are here and who are not with us tonight, but it's just an excellent webinar, and the whole idea and the whole premise and the whole work, and it looks like it feels like you're so passionate, enthusiastic. It's almost your calling to do this kind of work. So I would like to congratulate and thank you, your whole group, for that, and thank you for taking my questions, and really excellent presentation. I'm really applauding your work. Thank you.
Thank you.
Thank you. That's very kind of you. We
Yes,
appreciate it.
thank you very much.
All right, and we are a little past 9, so maybe we should wrap it up. And yeah, Larissa said it all, so that was good of her. Thank you, guys. You know, we do for Friday, if anybody wants to have their students participate and learn something here about the bottle biology and phenology at 1 o'clock Eastern time. We'll have a student-centered, using that word, student-centered webinar. But you need to register, and I think it was, okay, yeah, I can't share my, well, maybe I can share my screen. Let's see here. You can see I was reading my posts in my class. So this tiny URL is the student one to register for Friday. So please do that, so then we know how many classrooms may call in. And we've had nice attendance in the past. So anyway, I wanted to thank everyone for calling in tonight and for this great webinar on Green Up. And I enjoyed it. I learned a lot myself. Hope you did too.
And thank you for the comments on chat. I know we don't have time, but some people have some good ideas, so we appreciate that.
Yes, thank you. And Kevin, really quick about your butterfly bush. It's not really a bush. That's why it looks dead. It'll be fine.
Well, I guess I was thinking sometimes it grows buds, right?
Uh-huh.
Like a tree or a bush, but then sometimes that whole thing dies off and it'll start sprouting at the base again.
Yeah, so it's called it's a biennial sprouting bush. Well, it's not really a bush.
Right.
Yes, you're right. That those last for 2 years. You get 2 seasons, and then it all dies back and starts from scratch. So it's like a phoenix.
Okay, so I should cut that dead part.
Yeah.
Okay, thank you. That's what I was wondering about.
You're welcome. Sorry. Thank you
Awesome.
all for coming.
Thanks.
So bye-bye.
All right, take care.